Right. So whatever notes I make from this today, I will be posting a copy on the Canvas page after this. So you guys will have those notes, but feel free to take your own as we go along in case you need something in your own notes. But we're going to go over these basics today. I'm going to give a brief summation of our eukaryotes and our eukaryotes and our viruses. It's been a little while, so I want to make sure that we have our foundations there. Then we're going to do genetics, then the metabolism stuff, and then the immunology stuff. And if we have time, we will try to put it in perspective of the viral, bacterial, fungal, protozoa, and how much different kinds of infections and the disease progression, but odds are we're not going to get to that. We got kind of cut short with our immunology talk in my previous class, so I imagine that we'll get cut short in this class as well, just with time constraints there. But at the bare minimum, I will try to summarize MHC and the CD stuff at the bare minimum in that immunology stuff, okay? So to start off with, we're going to begin with our eukaryotes, our eukaryotes, and our viruses. And so first things first, we have all of our pathogens. We have the ones that are based on cells and the ones that are acellular. For our cell guys, we have our eukaryotes and our eukaryotes. For our acellular, no cells for these guys. These will be our viruses and our prions. These guys do not have a cell. No cell structure at all. So what's a cell? Does anybody know what a cell is? Anybody can define that for me? Are you sure? Right. Some cells don't have a nucleus. Do bacteria have a nucleus? Are they made of cells? Yeah. Right. So cells are membrane-bound structures. They have a membrane around them. All cells do have at least an outer membrane, right? They have at least an outer membrane and are able to replicate themselves completely on their own. That's a cell. So mitosis, for example, or binary fission, whatever it is that the organism uses to replicate, but they can make a copy of themselves. They are a membrane-bound unit that is able to make a copy of itself. That is a cell. Viruses cannot make a copy of themselves. They have to use your cell machinery. They have to hijack the components of the host. Prions, those are just misfolded proteins. So they are definitely not going to be just replicating themselves. They have to find properly folded protein and then cause it to misfold. So that's how they propagate. But cells, they have a membrane, at least one membrane around them, and they're able to multiply themselves. Okay, so eukaryotes, these include our bacteria and our archaea and our eukaryotes. This includes fungi, protista, which includes our algae and our protozoa. And then we also have our animals. And we also have our plants too. We didn't really talk about the plants, but our animals, this will include all animals like insects, people, and whatever. But for our microbes that we had talked about that cause disease, this includes our helminths. Yes, we also have plants, I know. I'm aware, but they didn't cause any disease, so we weren't as worried about them, right? All right. So this is our basic breakdown of all of our pathogens. Let's start with our eukaryotes. Also known as prokaryotes. Defined by, why is this doing this? Take off my, I think it's my bracelet. So I was going to take it off, but now it's like the most difficult thing I've ever done in my life, even though sometimes it just falls off. Okay, So our eukaryotes, also known as our prokaryotes, our defining feature of these guys, no nucleus. Again, this is our bacteria and archaea. No nucleus and no membrane-bound organelles. We'll talk about our membrane-bound organelles when we get into the eukaryotes. Okay, so no nucleus. So where do they keep their DNA? Just in the cytoplasm, exactly. Just in the cytoplasm. Everything in the bacteria and archaea either happen in the cytoplasm, or if it has to happen on a membrane, it happens on the inner membrane. Okay, so that's really the only places there are in the cells. And so most of it is going to happen either in the cytoplasm or on the inner membrane. And so this is basically what our cells will look like. Well, I'll draw them in a minute. I want to skip too far ahead with this stuff. So coming back to our guys, they have no true organelles. We'll talk about the organelles. I'm going to go over them in a little bit with our other stuff. So these are our bacteria and archaea. And something else that defines these guys are their cell envelope. What's the cell envelope? It consists of the inner cell membrane, and the wall. What is happening with this? Like, I don't understand why it is doing this. And if it has one, an outer membrane. What's the cell wall made of? What's the bacterial cell wall made of? That's the membrane. The membrane is made up of phospholipid bilayer, right? Why is it like this? Why is it doing this? Anybody know what the bacterial cell wall is made up of? Well, it might have some protein in it, but way more important than that. What'd you say? Peptidoglycan. You guys know this. I know you know this. I know you do. Peptidoglycan. What? How did the gram-positive stain work? Because this thick peptidoglycan wall trapped the crystal violet in there, right? We remember this, yes. So peptidoglycan is what makes up the bacterial cell wall. The outer membrane, not all of them have this. So where do we find the outer membrane? Only on what kinds of bacteria? Gram-negative. So that cell wall, that peptidoglycan cell wall, can also be decorated with that wax that we see in acid-fast bacteria. And that wax is mycolic acid, right? And we see that in Mycobacterium, right? So here's our basics of what are gram-positives. If it'll let me freaking write. I don't know why it's like this. Gram. I'm going to lose it. And I don't know why it's doing it either. Gram-positive and gram-negative. Here's our inner membrane. They both have an inner membrane. They both have DNA. Their DNA is in the cytoplasm. Not in a nucleus, right? They have ribosomes. It's just in the cytoplasm. And this is where their protein production and everything will be happening. And some other stuff is happening on their inner membranes and whatever else, right? Cool. So there's their structures for these inner membrane guys. They were both the same, the exact same on that level. Then we have the gram-positives have a thick cell wall. Whereas our gram-negatives have a thin cell wall. But our gram-negatives also have the outer membrane. And that outer membrane sometimes has some other structures on it that define the gram-negatives. This is also known as LPS, lipopolysaccharide, which what else do we call lipopolysaccharide? Endotoxin. Okay, we following? We caught up with bacteria? Any questions about the basics of bacterial structure anyways? So that whole thing, the inner membrane, the wall, and the outer membrane, if it has those things, all of that stuff is called the cell envelope. And then when we talk about the Graham's thing, it's actually identifying our organisms based on the cell envelope, more generally speaking, than it really is on the cell wall. Now, does it depend on the cell wall and how thick and thin it is? Yes, it does. But we're really identifying organisms based on their cell envelope. It's the better way of approaching that. All right. So that's the gist of the bacteria. Let's talk about our eukaryotes. These guys do have a true, yes, they do have a true nucleus. And they have organelles, which are membrane-bound functional units inside the cell. Here are some examples of organelles. The nucleus, the control center of the cell, it also houses the genetic material. The mitochondria. What's a mitochondria? Say it's a powerhouse. What happens there? Thank you. ATP production. Interesting. So we know that in eukaryotic cells. So our eukaryotes, these are going to be, for our microbes, this is our fungi, which includes yeasts. This is our protozoa. This is our helminths. Right? And us and other multicellular organisms as well. But for our microbiology stuff, it's these guys. All right, great. So we know mitochondria is the site of ATP production in eukaryotes. Where do bacteria make it? Either in the cytoplasm, that's where glycolysis will take place. Glycolysis in Krebs takes place in the cytoplasm. The electron transport chain, though, that happens on a membrane. And so in bacteria, where our electron transport chain is on the folds in our mitochondria, the cristae, remember? That in bacteria, that happens on the inner membrane of bacteria. All right, so it's an important one to remember. That's a difference there. What's another one that we have for an organelle? We have the ER, and also known as the endoplasmic reticulum. Endoplasmic reticulum. Most people can tell me that this is the site of protein production, and that's correct. But there's actually two parts to the ER, the rough and the smooth. What's the rough ER? Anybody know? It has ribosomes on it. That's why it's rough. It's the site of protein production. What's the smooth ER for? This is where we're going to have production of other biological macromolecules. So this will be lipids and carbohydrate production. Anybody in here know what the Golgi apparatus is for? Excellent. Golgi apparatus is our packaging and shipping. It's the Amazon warehouse of the cell. Okay, beautiful. We have other structures in the cell like lysosomes and vacuoles and stuff like that. A vacuole is basically just a storage vesicle. It stores like water or food or anything like that. Whereas a lysosome it is basically a vesicle that just contains digestive enzymes and toxic molecules that can help digest foreign material, including like pathogens. So we will merge the lysosome with a phagosome, and a zome is just a vesicle. And so the two zomes come together and make a phagolysosome in phagocytosis. And so that's basically what we're going to have in our innate response with phagocytosis, and how that's going to work is those vesicles will be merging together with the lysosome that has the digestive enzymes in it. Let me make sure I'm covering the stuff I want to cover. Chloroplasts. Chloroplasts also, we kind of talked about chloroplasts with the plants, but we'll add them on here. A chloroplast, another membrane-bound organelle. What happens in chloroplasts? Photosynthesis. Yeah. So can bacteria undergo photosynthesis? Not all of them can, but are there any bacteria that can undergo photosynthesis? Yes. And so if they're going through photosynthesis, where does photosynthesis happen in those guys? On the inner membrane. Instead of on the membranes of the chloroplast, on the inner membrane. Or you've either got inner membrane or cytoplasm. That's your only choices with bacteria, okay? So inner membrane or cytoplasm is all you have with bacteria. And the eukaryotes is where it gets complicated. What happens in the nucleus? What happens in the mitochondria? What happens in the ER? What happens in the Golgi? What happens in the chloroplast? And what happens in the cytoplasm? Because these eukaryotes also have stuff going on in the cytoplasm as well. So there's a lot more going on with the eukaryotes. All right, so let's move on to our next topic, which will be the viruses. Viruses are acellular, obligate, intracellular parasites. They rely on the host cell machinery to allow them to replicate. And all viruses have to have these two things: a genome of some kind. So that genome could be DNA, or it could be RNA. That DNA could be single-stranded or double-stranded. Same thing with the RNA. And that single-stranded version can be positive sense or negative sense. Positive sense means that a ribosome could just read it. Negative sense means we have to make a copy of it in order for a ribosome to read it. Basically, that's like the watered down version of it. All right, we'll talk about transcription and translation in the next part. But for now, viruses, all viruses have to have a genome. And they also have to have something else called a capsid. And a capsid is the protein coating on the genome. And it's made of parts called capsomeres. Some viruses some viruses might have an envelope. Envelope is the membrane covering stolen from the host cell. A naked virus means they don't have an envelope. So, poliovirus. Poliovirus is a virus that is a naked virus. It does not have an envelope. So I had a student in my previous section ask me, well, the ones that don't have an envelope must be easier to get rid of. And that is incorrect. The problem with enveloped viruses is that they actually require that they have the envelope. And that means they have a membrane structure around their surface. And that membrane is susceptible to surfactants and things like that that we often use in cleaning. It's also susceptible to drying out. There's usually a moisture component in there as well. So enveloped viruses are the weakest, generally speaking, of all of our pathogens in our control methods. So envelope, because they have to have that membrane. Non-enveloped viruses, also known as naked viruses, like polio, are actually hardier because they don't have to have that membrane. That membrane is going to be more susceptible to being broken apart and things like that. Right. Some viruses also have special proteins that come with them. That they package in the virus particle. This could include, for an example, HIV's reverse transcriptase. All right, what else am I wanting to add in here? Oh, I skipped the helminths and that stuff. Okay, well, whatever. We're on the viruses now, so let's finish up the viruses. Yes, okay. So that is the basic gist of it. And so all viruses use the host cell machinery to replicate. And that's what makes it a parasite, because it's using our resources in order to multiply itself. All right, come back up to the eukaryotes because I wanted to add in some stuff on that so I don't leave you guys out on that. For the eukaryotes, the fungi. All right, so the fungi first. This includes our yeast and our molds. Yeasts basically bud off in round structures from one another, whereas molds have those hyphae structures where we have the branching off and that sort of stuff. So that's the difference between those guys. The fungi are multicellular or single cellular. They're typically going to be, most of them are multicellular, but they can be single cellular or multicellular. They are not able to photosynthesize. They do not have chloroplasts. Okay, no photosynthesis for these guys. They do have a cell wall, usually made of chitin or cellulose. What else about the fungi? They can reproduce asexually or sexually. Asexual reproduction is the one that we talked about, really. All right, our asexual reproductive structures for these guys, there's two different ones, the covered and the not covered, the open. The open asexual reproductive structures are called conidiospores. Organisms that are able to make these structures are called conidiophores. And then so the ones that have the covering, so this is the ones that we looked at with like penicillin. They look kind of like this, or it might look like a dandelion, but it's got like all that stuff coming off of it. But if it's got the cover on it, it would be like a dandelion that had a cover around it and you'd be able to see it in the image. These are called sporangiospores or sporangiophores. All right. The Protista kingdom included our algae. These guys are photosynthetic. They have chloroplasts. These guys are our primary producers of oxygen on the planet. And then we had our protozoa. always single cell, our protozoa. I don't know why I put two L's in always, but it's whatever. Always unicellular. All right, so we had 4 major categories of our protozoa based on motility. We have 4 motility types. This is going to be the ones that move by flagella. This would include Giardia and Trichomonas and other ones, but these are just examples. We have the ones that move by pseudopods. The fake feet, these are also known as the amoebas. This would include things like Naegleria fowleri. Or Acanthamoeba. Both of these guys, a source of meningoencephalitis, the brain-eating amoebas. What kind of disease does Giardia cause? Anybody know? Chronic diarrhea, chapter 23, right? Chapter 23, Giardia, chronic diarrhea. Greasy, malodorous stools with heavy flatulence. We're talking diarrhea for over a month. Trichomonas, what kind of disease does that cause? It is an STI. Yes, so it's a protozoal STI. It is the most common non-viral STI by far. So it comes in 3rd place with all of our STIs. Our 1st place STI will be HPV, human papillomavirus. Causes genital warts as well as cervical cancer. Just because it causes cervical cancer does not mean it causes genital warts. They do not come hand in hand. In fact, most people that have HPV will not have any signs or symptoms, and most people will clear it from their bodies naturally. That's why Pap smears are so important, because we want to be looking for any abnormal cells that come as a result of HPV infection. Over 90% of sexually active adults will have had HPV at some point. Over 90%. Most common STI. Next is herpes. That shouldn't be that surprising. 1/4 of all sexually active adults will be positive for herpes simplex 2, the genital herpes. 1/4. Most people will have had HSV1, which is the one that causes oral herpes, where we get our cold sores from. We're talking over 90% of people will have had that. That's just common. But you can have genital herpes in the mouth, and you can have oral herpes in the genitals. They can cross over. It's just more commonly the oral herpes is HSV1, and more commonly the genital is HSV2. So that's those are the 2 most common viral ones. And then we have Trichomonas vaginalis, which causes, trichomoniasis. If you guys get to going into the ER, you do your rounds in the ER. If you guys do a round at the ER at Southwest Integris, 100% chance you'll find hookers that have this. They're coming. I mean, I'm telling you, I worked in a lab there all the time. All the time, the hookers have this. Okay. All right. Moving on. Our other guy, the ciliated cilia. Some of our protozoa can also move using cilia. Little tiny oar-like projections that help with moving the fluid. We didn't talk about any specific guys that have cilia that cause disease, but there are some, but I'm not going to test you on it, so I'm not going to go over it. Okay. Cilia. And then our last one are our non-motile, which are known as apicomplexans. And this includes diseases like malaria. There are other ones too, but this is an obvious one, I feel like. All right. So that's our protista. And then we had the helminths. These are all animals. These are our parasitic worms. So I'm going to add another page here so I can cover my helminths well. These guys are all multicellular and have several life stages. This will include forms that are like egg forms, larva forms, as well as adults. All righty. Our helminths. That basic like parasitic worm term basically divides into two categories: the flat and the round. The flatworms are known as platyhelminthes. And the roundworms are known as, nope, nematodes. Getting ahead of myself. Nematoda. All right. The platyhelminthes are separated into two categories. These will be the tapeworms and the flukes. Tapeworms are known as cestoda, and the flukes are known as trematoda. Some examples: roundworms. This would be things like the giant intestinal roundworm, like Ascaris lumbricoides. This could also be our pinworms, which are our Enterobius verbicularis, and that sort of stuff. I'm not going to give every single example and categorize them right here, but these are some examples of these. For our flukes, some example of some trematodes. This would be like our schistosomes, our blood flukes, or our blood flukes. Our liver flukes, those were Opsithorchus sinensis and Clonorchus sinensis. Those are our liver flukes, the Chinese liver flukes. These are all chapter 23 things. And then our cestoda, our tapeworms, this would include things like Taeniosolium, and there was like also Diphyllobothrium latum, which was the sushi tapeworm. And those, I'm not going to write that one out. Sorry. Don't cry over that one. All right. So now we've done all of our basics for our pathogens. Now we went over our eukaryotes. We went over our eukaryotes and even broke them down. And we even talked about our viruses. So now our next step is going to be, oh, I talked more about the I talked about lysogenic stuff. I'm imagining you guys want to go over that too. All right, coming back to our viruses, we know that they're going to require our cell in order to replicate all of their parts. Let's talk about lysogenic versus lytic cycle. First of all, bacteriophage is a virus that infects bacterial cells. All right. And then these terms, lysogenic and lytic cycle, apply to bacteriophages only. Okay. So the prophage, when the viral phage DNA, phage genome, is inserted into the bacteria's genome. All right. So the lytic cycle happens whenever the virus is replicating making new virion particles, and it bursts out of the bacterial host cell. That's a process called lysis. The lysogenic cycle happens when the prophage inserts into the bacterial genome and it hides and just waits, right? If a prophage imparts new genes to the bacterial host by inserting itself that way, that help with survival of the bacteria, or help cause disease in the host. This is a process called lysogenic conversion. When the bacteria gets new, helpful, beneficial traits that we would call virulence factors. Virulence factors are any traits that help the bacteria cause disease, help any pathogen cause disease. A virulence factor is anything that helps a pathogen cause disease. And so if the bacteria gets a virulence factor from being infected with a phage that inserts new genes into its DNA, that's called lysogenic conversion. Do not think for a minute this is rare. Almost every single pathogenic bacteria is pathogenic because of lysogenic conversion. So the toxins that strep throat makes, lysogenic conversion. The toxins that cholera makes, lysogenic conversion. The only reason that cholera even causes disease is because of the toxins that it got from a phage that it was infected with. Pretty, I mean, a little bit meta thinking, but it's pretty significant. All right. All right, now let me make sure that we're going on the right path. We are. Let's get into genetics. We're going to start off with DNA replication. This is a process for replication, a process of making copies of DNA. So DNA is what we call anti-parallel. The backbone of the DNA molecule runs in opposite directions on each strand. So this is where we have one strand running from the 5' to 3'. The other one runs in the opposite direction. All right. And DNA replication is semi-conservative. Does anybody remember what that term means? Each new DNA molecule consists of one old and one new. So this means that if this is your old strand, we use the old strand as a template to create the new. And this is based on that concept of in DNA, your nucleotides are A, T, C, and G. A binds with T, T binds with A, C binds with G, G binds with C. All righty. And so now whenever we're going through DNA replication, make sure that I'm covering it all, just some conservative, old and new. Yes. Okay. So whenever we go through DNA replication, then this top strand that is continuous, what's that one called? This is the leading strand. All right, so if that's 3, then this is 5. And since DNA replication can only happen in the 5' to 3' direction, it's unidirectional, then whenever our DNA polymerase comes to add on, it'll add on like a little fragment here. Start it here, and we'll add on another little fragment here as it opens up more. These are the fragments that are made. What are those fragments called? Okazaki. Okazaki fragments, and these are on the lagging strand. All righty, so DNA polymerase is the, not what I meant to do. DNA polymerase is the enzyme that's going to be making all of this happen. So this is DNA polymerase, and it always operates in the 5' to 3' direction. And then whenever we have these Okazaki fragments, we'll have to seal them together. What enzyme does that? Do you guys remember what it's called? This is called DNA ligase. It seals the Okazaki fragments together so that it's one continuous strand. If we just left them as fragments, your body wouldn't be able to deal with it. It couldn't make your genes, nothing. No transcription, no translation would ever occur. So that's DNA replication. Any questions about that? Semi-conservative. We'll have new strand will be completely one old and one new. We use the old one as the template. And we do that as we open up the DNA. We make in the 5' to 3' direction, going in towards that replication fork as it's opening up. That's the leading strand that's made continuously. The one that's made in fragments and segments is called the lagging strand. And those fragments are called Okazaki fragments. And we seal them together once it's done with the DNA ligase enzyme. Reforms those bonds. So it's one continuous strand. All right. Moving on to transcription. Transcription is the process of using a DNA template to make RNA. So if your DNA, your nucleotides were A, T, C, and G, your RNA is going to be A, U, C, and G. What's the U stand for? You guys remember? Uracil, exactly. Whereas A, C, and G are always the same. A is adenine. C is cytosine. G is guanine. And then T for DNA is thymine. Okay. All righty. So if your DNA sequence, let's say that this was your DNA, and our DNA sequence read Just give it 6 because I don't want to do too many. A, G, A, C, T, T. Then the opposite strand would be T, C, T, G, A, A. And if we are going, this would be what we would be doing for replication. But if we are going through transcription, we would have a DNA strand that might be that A, G, A, C, T, T, and then the RNA would use that as a template to make our RNA, which would correspond to it, which would be U, C, U, G, A, A. All right, we have 3 different types of RNA. We have mRNA, which is the working copy of a gene. We have rRNA, which constitutes the structure of a ribosome. And we have tRNA, which is our transfer RNA, which transfers an amino acid based on the mRNA codon sequence. All RNA, regardless of what type it is, was coded for in your DNA. And DNA became RNA through transcription, always, no matter what type of RNA. The RNA came from the DNA in your genes through transcription to lead to that. So transcription itself has 3 steps. Initiation, elongation, and termination. In transcription, where does initiation start? Does anybody remember what that site is called? So I'll help you out. RNA polymerase, which is the exact same thing as DNA polymerase. It's just using RNA nucleotides instead. All right. So RNA polymerase binds at a site called the promoter region. In elongation, we are going to add RNA nucleotides complementary to the DNA template strand sequence. This will happen in the 5' to the 3' direction, always. Then termination is just where we stop that. There's a terminator sequence for that, but I'm not going to worry about that. I'm not as worried about that part of it. Okay, so that is when we make our RNA. So we've gone through replicating our DNA. We've gone through making our RNA. And now we've gotten to the big bag, the whole point of it all, which is to make proteins. And so translation is where we take our RNA and turn it into protein. The things that help with that, the things that feed into that, are going to be our ribosomes and our tRNA. All right. So translation, basically your mRNA is a sequence of nucleotides that occurs in triplets. On the DNA, that same exact genetic code on the DNA, we actually literally call triplets. But on the mRNA, those are called something else. Does anybody remember what they're called? Y'all are so much quieter than my other classes. Like they are like shouting out answers, whether they're right or wrong. This is where I get worried about you guys, right? Because you guys don't talk about this stuff enough. So what is this called? The 3 nucleotide sequence on an mRNA that codes for an amino acid. What's the term for that? Codon. Thank you. Codon. And 1 codon will code for 1 amino acid. How is that? Because the codon that codes for the amino acid is recognized by the anticodon on the tRNA that carries that amino acid. This is how this will happen. We have our ribosome, and then we have our mRNA. The ribosome clamps on to it, whatever. All right, and then the tRNA, again, codon. This is not my best artwork, but whatever. This is the tRNA, and this is the anticodon. And that tRNA is attached to an amino acid. And the next one will come in and it'll join the next amino acid on it, and so on and so forth. And that this whole process is translation. So that whole thing that's going to make, we would call it a polypeptide. And that may or may not make a functional protein as a result. So this will kind of remind us back to the structure of proteins. We have the primary, secondary, tertiary, and quaternary structures of proteins. Make sure I'm covering everything I want to on translation. Okay. These have the same 3 steps as previously before I talk about the structures of the proteins. Steps, initiation, where the ribosome is going to bind on and start and add the first one is going to be the AUG codon, coding for methionine, always. The second is going to be elongation. Just continue to add on the new amino acids to the existing polypeptide. According to the codons sequence. And the third is going to be termination. This is based on a stop codon, also known as a nonsense codon. These guys do not code for an amino acid, and because of that, it just causes the ribosome to dissociate and drop off and let go of the polypeptide that it's making. Protein structure. Yes.
Transcription steps and the termination for that one.
Mhm.
Is it also a stop codon or is it just a stop?
No, there's a terminator sequence for that one. So that's a terminator thing. So it's not a stop codon for that one. Stop codon only dictates the stop of the translation.
Okay.
Yeah. So the stop of transcription will be dictated by a terminator sequence. We don't need to know anything else about it besides that, though.
Okay. Thank you.
Yeah, good question. All right. So just to remind you guys, since we're making proteins now, I'll just remind you guys of protein structure. The primary protein structure is just the amino acid sequence by itself. The secondary protein structure is going to be how the different amino acid groups right next to each other, amino acid group neighbors, cause structures like alpha helices and beta sheets, beta pleated sheets. So these are the guys that are immediately next to each other. In tertiary, we now have groups like from far away. We have hydrophobic, hydrophilic, polar or non-polar, and even some like covalent bonds being created. At the large scale, on a single polypeptide. So this is that step that I called like the tying of the bow. Quaternary structure is when you have multiple polypeptides come together to create the functional protein. All right, so that brings us into our enzymes. An enzyme, its whole entire functional structure if it is a conjugated enzyme, meaning it has multiple parts, that whole entire structure is called a holoenzyme. The protein portion of that called the apoenzyme. This is where we will find that active site for binding the substrate. The non-protein portion is called the cofactor. This can be inorganic, like we see with metal ions. In this case, we just call this just a cofactor. But if it is organic, meaning that it has carbon hydrogen backbone, that this is called a coenzyme. And that most of our vitamins actually fall into this because those are going to be organic structures based on carbon hydrogen backbone, and that they are required for our protein to function, then they are a coenzyme. And this includes things like NAD and all that we talk about in metabolism. Those are organic. Yes.
I just want to make sure I understand this right. So the non-protein portion is either a cofactor or a coenzyme, depending on if it's organic or.
So it's always called a cofactor.
Okay.
And if it is organic, it is also called a coenzyme. So a coenzyme is a cofactor.
Okay.
But we just will define it even further as a coenzyme. Okay. Good question. Make sure I'm covering this. We had 6 basic enzyme groups and 4 of them that I decided that were actually like really important. And so I'm going to go over those groups, enzyme groups. These are going to be our oxidoreductases, our transferases. So oxidoreductases are involved in electron transfer. Transferases are involved in moving molecular groups from one molecule to another. And then we had our hydrolases. These guys are going to break water. To break molecules apart. And then we have our ligases. These guys make water to make larger molecules. Kind of the opposite of the hydrolases. So you guys remember how in lab, we talked about the urease, the starch, and the gelatin tests were all hydrolysis tests. They test for hydrolysis of starch or hydrolysis of urea or hydrolysis of gelatin. Those enzymes, urease or amylase for starch or gelatinase, were all hydrolases. They were all enzymes that were involved in hydrolysis. So we were breaking apart either starch, gelatin, or urea by breaking water in order to break those apart. That's what hydrolysis is. So we've actually been doing this stuff that probably you guys were stored in your head completely separately from when we talked about chapter 10. It's the same shit. Okay. Same shit, different day. And breaking that stuff down. So there's that stuff. Also different kinds of ways that we can regulate enzymes. This will be either competitive or non-competitive. The difference is in competitive enzyme inhibition, the inhibitor binds at the active site where the substrate normally goes. In non-competitive inhibition, the inhibitor binds at the allosteric or regulatory. I can't spell right now, apparently. Regulatory site. Basically, not the active site, somewhere else. The active site is where the substrate binds. Okay, so we remember how just a minute ago we talked about oxidoreductases. What is a redox reaction? Well, in a redox reaction, we have transfer of electrons between molecules, right? One gains an electron and one loses an electron. Oxidoreductases are enzymes that help in redox reactions. All right. So what is oxidation? And what is reduction? Lose electrons, right? Lose electrons, gain electrons. And so you guys can remember this from either Leo says grr or from oil rig. We went over these previously, but Leo says grr, lose electrons oxidized, gain electrons reduced, or oil rig, oxidation is loss, reduction is gain. So either one that helps you remember it. And this is literally one of those things that I used to have to like write down when I was doing like redox problems for chemistry, that I even had to have a helper with that. So that's what's happening in redox reactions. Okay. Good. Now we're on track. Okay. Aerobic respiration. We have 3 steps: glycolysis, Krebs, and the electron transport chain. In glycolysis, the whole point of glycolysis is to take a 6-carbon glucose and turn it into 2 3-carbon pyruvates. In the process, we'll make 2 net ATP. We'll also make some NADH, but we will have no CO2 made. In this intermediate step, we will turn pyruvate, which is 3 carbons, into a 2-carbon acetyl CoA. In the Krebs cycle, we will cycle that acetyl CoA in a big old circle. As we do that, we're going to kick off several carbon dioxide molecules. We're going to be making a little bit of ATP, but that's not the whole point. The point of this whole thing is to get a whole bunch of our electron carriers, NADH and FADH2. This is the whole point. The point of glycolysis was to make pyruvate. The point of Krebs is to make NADH and FADH2. Is this the first time in all of this process that we made carbon dioxide? No. Because we make it whenever we go from pyruvate to acetyl-CoA. That's the first time we make carbon dioxide. Then we make it again in the Krebs cycle. But that is the last time that we make carbon dioxide. We don't make carbon dioxide in the electron transport chain. So the electron transport chain, you're basically going to have a set of these proteins in a membrane. And you'll have this other set of proteins down here we call ATP synthase. All right. So what happens is NADH or whatever BS that you made to carry your electrons will give off an electron and an H plus. The electron shuttles its little self through these proteins here. Anybody remember what these proteins are called? They're called cytochromes. Anybody remember where that sounds familiar from? Maybe the last lab on respiration with oxidase. Cytochrome C oxidase is the last one in the electron transport chain in aerobes. It will be this one right here. Okay, so whatever though. So there's our electrons, and then these H pluses all get pumped out while those negative electrons move across the cytochromes. The positive H pluses get moved outside of the cell. We create this gradient. This is known as the proton motive force. And then this causes the protons to move back in. And then we have the electrons that come off too. So the electrons plus the H pluses plus, oh, come on now, half of the O2, leads to producing water, which is how we maintain our gradient going. And then whenever the protons come through, we are able to take ADP and then turn it into ATP. This is the point of all this, is to make ATP. This process is so important and relies so heavily on this oxygen to help maintain this gradient that when I suffocate you, you die within minutes. Yes, because you ran out of oxygen. The only thing you need oxygen for is to make ATP. So what you really die from is running out of ATP. When you run out that fast, within minutes, your body will run out of ATP. So that's pretty impressive how much your whole body is actually relying on ATP and how much aerobic respiration plays into making your ATP. Now, anaerobic organisms, what's the difference? They don't use oxygen as their final electron. So they don't use oxygen at all. So they'll use something like nitrate or a sulfate compound, and they will reduce nitrate or sulfate into some nitrogen product, like we learned about nitrite or something else, or they'll reduce sulfates into H2S. Instead of H2O, they get H2S. That is sulfur reduction. That is why we had black precipitates in SIM and TSIA, anaerobic respiration. So there's one other kind of anaerobic respiration that we call the organisms that perform it, we call them facultative anaerobes. Does anybody remember what that kind of respiration is? What it's called? It's called fermentation. Okay, so facultative anaerobes do not, if they're able to do aerobic respiration if oxygen is available. If oxygen isn't available, fermentation stays in glycolysis. They don't go on to Krebs or electron transport chain. And so the only way they're making ATP is in glycolysis. Only 2 net ATP. So if you go through aerobic respiration, the net ATP will be 38 for each glucose molecule. Okay. Alrighty. Now I think we're in the immunology stuff. Yes, we are. Okay. No, I want to switch back. Immunology. What are white blood cells called? What's the correct term? Leukocytes. And we have 2 categories of the leukocytes, the granulocytes and the agranulocytes. What are our granulocytes? The fills and the mast cells. This is real basic, right? But you get it. Well, our granulocytes are our lymphocytes and our monocytes. The lymphocytes are our T and B cells. The monocytes can be moved into tissues. So the monocytes are in the blood. They move into tissues and they become macrophages or dendritic cells. The fills, there's 3 different neutrophils: basophils, eosinophils, and neutrophils. And we'll also bring us to our phagocytes and our antigen-presenting cells. Now, leading into our acquired immunity. So our phagocytes, we have neutrophils, macrophages, dendritic cells, and also monocytes. Because these guys are monocyte derivatives. The monocytes are in the blood. Once they move into the tissue, they become macrophages or dendritic cells. So they're all the same thing. Our antigen-presenting cells are monocytes and their derivatives, as well as B cells. So B cells are not phagocytes, and neutrophils are not antigen-presenting cells. So you see our go-between here is really our macrophages and our dendritic cells, monocyte derivatives. All right. So having said that, we're going to go into, this is our last stretch here, okay? We're going to go into our acquired immunity. Those are antigen-presenting cells. So this will get us into what even are antigen-presenting cells and their function and everything like that. But first, we have to understand what MHC is. MHC stands for major histocompatibility complex. What's the function of MHC? Close. MHC is a very general molecule. It doesn't have a specific interaction. I don't care what antigen you are at all. But what MHC does, it presents, no matter what kind of MHC, it doesn't matter. All MHCs present antigen to T cells, period. That's what MHC does. That's its function, is to present antigen to T cells. There are 2 types of MHC, MHC1 and MHC2. MHC1. Is this what we're doing now? MHC1 presents, we know this part, presents inside antigen. Whereas MHC2 presents outside. MHC1 only recognized by CD8 positive T cells. And MHC2 is only recognized CD4 positive T cells. CD8 T cells versus CD4 T cells. MHC1 is on all cells, all nucleated cells, was pretty much all cells. The defining characteristic of MHC2 only on antigen presenting cells. Right? CD8 is a molecule that recognizes MHC1. That's what it does, and it's the only one that does it. CD4 is a molecule that recognizes MHC2. That's it. So what recognizes the antigen itself? This is the T cell receptor. All right. So that if we do this, basically, here's what happens. We have our antigen presenting cell. We'll say that this is a macrophage, okay? Macrophage. And it is going around and it's a blobby guy and it's eating some stuff up and it picks up, get out of here. It picks up this crap from outside. It goes through phagocytosis and then puts it on, because it picked it up from outside. What is it presenting it on? MHC2. It's from outside. All right. Then our T cell receptor on our actual T cell. Recognize that. Now, if we're interacting with MHC2, what molecule will interact with MHC2 itself? CD4. There we go. So if this has CD4 on it, what type of T cell is it? We know it's a T cell, but what kind? T helper. Then this will allow these guys to cross-activate. And then that leads to this becoming TH1 and TH2. TH1 sends activation signals for inside stuff. TH2 sends activation signals for outside stuff. So then what I'm saying here is that now suddenly you have an infected lung cell that is infected inside with a virus that it'll express out on its MHC1 because it's coming from inside. Inside antigen presenting on MHC1, any cell can do that. Then next, our, let's put them in a different color, T cell receptor on, if this is MHC1, what molecule interacts with MHC1? CD8, exactly, CD8. So this is a T cell also, because T cells always respond to MHC, but what kind of T cell has CD8 on it? Cytotoxic, right? We say killer, that's fine too. Cytotoxic, CD8 positive T cells. And these cannot act until they get this signal here from the Th1 cell. So you guys kind of see how they're relating now a little bit better? Okay. Did that help at all with that immunology stuff? I hope that it did. You guys can rewatch this. I will post this video and I will post these handouts or whatever you want to call them on Canvas for you guys. So you can refer to, I know you made your notes too, but hopefully this will help with clarifying some of that stuff. I feel like breaking down MHC is always just a molecule that presents to T cells. Just knowing that basic thing. And then you can divide it up onto which T cell is responding to which MHC. But keep in mind that T cells themselves are responding to the MHC that's presenting antigen. The T cell receptor sees that specific antigen, and then either CD4 or CD8 recognizes which MHC it is. And that is going to help drive whatever response comes after that. Okay. And for your B cells, just because I didn't draw it on here, but your Th2 cells, activate your B cells to turn into plasma cells. And what do plasma cells do? They make antibody. Remember that our B cells, I'm going to show you the picture for this one. I'm not going to draw it right here, right now, but with our B cells down here in the bottom of this picture up here, your B cells have a receptor on them. They aren't phagocytosis. They don't go through phagocytosis. How do they see their antigen if they're not phagocytosing the bad guy? They have a receptor on their surface. That receptor is called IgD. So don't forget that IgD is the B cell receptor. That's how B cells engage with their antigen. And they bring that receptor inside and express that antigen on MHC2, or they can interact with the T cells. The activated Th2 cells can also send the signal for activating the D cells to become plasma cells. The IgD is their receptor on their surface where they first interact with the antigen. The first soluble antibody that they crank out is always IgM. Then they will undergo class switching to either IgG, IgA, or IgE. IgG is your workhorse in your bloodstream. That's the important one for your memory. IgA is your mucosa one, and that we call the secretory IgA antibody in your mucus. And then IgE is the one that's associated with allergies or responding to your parasitic worms or protozoa. And that is it. And you guys are caught up with everybody. I will copy and paste this onto our notes and paste it onto Canvas for you guys. Yes.
Is the finals 100?
Yes.
Okay.
Yeah.
You said half of it would be over this last unit, right?
Yes, half of it will be unit 4 and half of it will be units 1 through 3, roughly. Don't bank on it being exactly 50-50 questions, but roughly that, because there will be some crossover questions too for that. Yeah. Let's copy this one. This page.
Thank you.
Yeah, and we'll go over the meta questions on Wednesday. We'll really get meta with everything, and try to relate everything back to itself. Some of them, yes, but we'll have critical thinking questions on there too. So those will be NCLEX style. Some of them will be NCLEX and intentionally